All right, so once again, hi everybody. Welcome in the first in-person / virtual class of BIOD25. And I'd like to start with a couple of general business before we go to today's class. The first is that during the break, we're going to have an external speaker, Kyle, so in the back of the room here, is going to give us a short overview of the tips and tricks that he learned last year about preparing the blog post. So Kyle is an ex-student of BIOD25, and he really liked the blog post, the act of writing. He even wrote 2 blog posts, and so that makes him the most experienced blogger in this class so far. So I think he will have some very nice tips for you guys. So just a few things that I need to discuss with you, especially because we're coming back in person. So I got the COVID update from yesterday, and these are a few things that I think are important for you to know and to discuss. So, as you know, we're back in person, and as you know, the government of Ontario has announced that they will remove the mask mandate by the end of March. So I wanted to discuss with you just a little bit some details about the mask, about how they work, and what is my opinion about them. And so the first thing is that so the masks, not everybody seems to be aware of how they work. You know, I can see some comments even from the politicians and the medical doctors in the media who say, I hope that people will keep their mask if they're vulnerable. But this is not the way the masks work. The masks don't protect you at all, almost nothing. The masks, when they're on your face, protect other people. So when I heard that, I thought, you know, how does that possibly, how can that possibly work? You know, it's the same one way or the other. So if they protect from the way out, they also protect on the way in, right? That's at least what I thought. In reality, the way they work is that when you speak, when you breathe, you produce some aerosols, they're called, and this says on these aerosols that the viruses are. You don't really spit viruses at all. You know, they weigh like, it's not like the air is full of viruses. They're on tiny bubbles of water, and these bubbles of water are going to decrease in size very, very rapidly. And so as you breathe, when they're exactly out of the nose, out of the mouth, they have a relatively big size, but within a couple of seconds, they're going to shrink to a very small size. And this is why the masks work, is because if I filter them when they're still big, then they're filtered by the mask. After, let's say, half a meter, 30 centimeters when they escape my mouth, they're already much smaller and they cannot be filtered anymore by the mask. So this is why this is asymmetric. If I have my mask, I protect you, but I'm not protecting myself. I have 2 masks. I've got an FFP2 here, and I've got this one, which is not FFP2. But already, as you can hear, it's difficult for me to speak even with a non-FFP2 mask, and it would be totally impossible with an FFP2. So for everybody's sake, I'm going to keep this relatively light mask, but I really invite you, as a matter of citizenship, let's say, to keep your mask on. You're not doing it for yourself, really. You're doing it for everyone else except yourself. So not wearing your mask during the classes is a statement that you're making against everyone. It's not a statement that you're making about, I don't care whether I'm infected because it's my decision. That's for sure is your decision, but that's not what the masks do. That's not what they're for. So that is why even when masks will not be mandatory, we're going to keep the masks in class. Okay? So why is that? It's also because I checked yesterday what is the state of the epidemic. And there's a bit of a disconnect between what you see in the media and what you see in the data. So we're all very happy that the Omicron wave is washing off, and that's fantastic news. So if you compare what it is today from what it was yesterday, it's much better. And this is why everybody feels kind of relieved, etc. By contrast effect, we feel much better. But let's look a little bit at other ways to see the data. So this is here the wastewater signal. So that's the amount of coronavirus you filter in the sewage. And people are doing this regularly, and you can see that we're now at that level. They were not even indicating the unit on the y-axis, but it doesn't matter very much. What matters is that it's not going down. Okay, you can see that actually we're way higher now than we were in December, at the time when we were not discussing whether we should remove the masks. Okay, so the idea of removing the masks now and saying that we're going to roll back to whatever was before in 2019 is not very consistent with what was happening in December. If we said in December we're not ready yet, then now we're less ready than in December. It doesn't feel this way just because in January it was bad and now this is better, that's for sure. But when you look at a comparison between December and now the epidemic is actually stronger. And it's not going down very, very fast. I can't speculate as to what the reasons are, but it's not going down very fast. Another very important point to consider when we make this sort of decision is, of course, the hospitals, because the death rate with this disease is not very high. It's the hospitalization rate that is much higher, and that is very much the problem. So when you look, there was this very nasty Omicron wave here in December and January, and thanks God we're really washing it off, and the hospitals are getting less saturated. So in black you can see the number of people in the hospitals in Ontario, and in red these are the people who are in ICU beds. And you can see that, yeah, we're going down very fast, and that's good news, but we're still way higher than everything that has been during the whole fall semester. Okay, we're at a point that is significantly worse now than it was in fall, when we were not discussing about opening that much. So this is why there are political and economic reasons for wanting to lift the restrictions. But when we look at the scientific facts, this is not consistent. Let's say if we say we're ready to do it now, we should have said we're ready to do it in September, for instance. Okay. And the last thing I wanna say about the masks, etc. I guess the guys online are getting bored because they don't care about these things. The last thing I want to say about the masks is that it's annoying. Okay, it's very annoying even for me, especially for me, I'd say, because I have to speak. But it doesn't cost jobs. It doesn't cost, isolation, etc. So in a sense, among the things that we can do, the mask is the one that has the least amount of negative effects, except on talking, of course. But in terms of the effects for everyone else on the society, they're much milder. And actually, this is one of the most efficient. That divides by two the or not. So dropping the mask at that point seems to be more driven by political motivation than by scientific motivations. That's all I wanted to say. That said, Ontario is going to say, these are the rules, and I will not be able to overrule them. I just wanted to give you a little bit of a more scientific view on that topic. Another thing that I'd like to discuss very rapidly is the midterm and the quizzes. So I've got the grades of the midterm. I'm going to release them very soon. You guys did actually very well. So I was very pleased to see that the grades are kind of nice, which shows that you have understood what this was about, and that's actually very nice. So congratulations, everybody. You'll see the update very soon. For the quizzes, so by now some of you have realized that there is something fishy with question 13, is that I think it's pretty clear that it was always the same problem. There were two versions of question 13, one in which you had multiple options and one in which you had no multiple options, and so you had to come up with the answers yourself. So what the heck was going on? It is one hypothesis that I'm trying to test. And the test now is finished. The last test was yesterday. And it has to do with the fact that I noticed in the previous year that whenever I ask questions that are quantitative with multiple choice, it looked like students are not engaging in quantitative methods. The way they solve the problems, was more by guessing, more or less randomly, but not really randomly, more by the who wants to be a millionaire approach. Eliminate two, because they look completely impossible, and then the last two, well, go by gut feeling, something like that, which is nice. I mean, that's what I do when I have a multiple choice question, but that's not what's really teaching you anything. And so I wanted to test that, and so the test was that the four options that you get, when you get the four options, were actually designed to hide the true answer from the rest. So if you went random, or if you even use your intuition, you were less likely to choose the right answer. And my observation so far is that indeed, when I design the questions in such an evil way, the grades were higher when you did not have the options. So that was just to confirm that indeed just giving you the options is actually very much an inhibitor of your learning, because it will favor a strategy of acquiring the answer, which has nothing to do with doing the exercise, actually. And yesterday this went pretty bad, let's say, in the sense that many people have copied the answer from those who had them, because there's a lot of passage now between the people who do the quizzes. And because the questions were so much designed so that you would not get the right answer if you do that, then yeah, very few people have answered the question yesterday. And many of them have kind of copy-pasted the answer from the other group, which was a bad choice. Anyway, so that's what I wanted to say. The experiment is over, and I've got the information I wanted. So sorry that I did not tell you, but I could not tell you because otherwise there would not really be research. But I've got pretty much what I needed to make progress on my teaching. So now we're going to finally start the lesson of today, which is about metagenomics. We're going to go until the break, and then after the break, Carl is going to give us his overview of the tips and tricks on how to do a great blog post. And next time, I'm going to give more information about how to write the blog post, how to read the papers at least. So we're going to start by just an overview of life on Earth. So first of all, what are the most common types of life on Earth? It really depends on how you define it, what is the most common. But if you go by mass, then you got a picture that looks like that. So here you can see the mass in terms of carbon of different types of organisms. And the organism that is the most abundant in terms of mass on the planet are plants. They represent by far the majority of organisms. But you also have archaea, bacteria, protists, like these unicellular animal-like organisms, fungi, and animals. So you can see that animals are actually fairly small. And we could think, yeah, among animals, we humans are so many that we must weigh quite a lot on the planet, right? Well, no, not at all. Here's the zoom in of the animal world, and you can see that most of the animals in terms of mass are arthropods, so spiders and insects, also crustaceans, spiders and insects, sorry. And we humans, we are here. Okay, so we're big. We're of course the biggest of all the mammals, but we're still nothing compared to the rest of the animal sphere, even though we're so many. So we're not that abundant in terms of mass on the planet. We're just a tiny spot on that relatively small corner here. Microorganisms are an important part of the biomass on Earth. Viruses have a very small contribution to the biomass, but they have by far the largest amount of individuals on the planet. So viruses are, here. It's just a dot on this very big square here, but they represent a huge number of individuals, of course, because they're so small. But the point is that most of organisms on the planet are plants, and second, those are bacteria. So we haven't been speaking a lot about prokaryotes so far. We've been speaking only about eukaryotes. So let's talk a little bit about bacteria and archaea. Prokaryotes consist of bacteria and archaea. They usually have a single circular chromosome with one origin of replication. Their genomes are rich in genes and poor in repeats. The genetic code is universal, but synonymous codons have different frequencies between kingdoms. What that means is that if you stick a gene from humans into a bacterium and you remove the introns, it's going to be translated, but it's going to be translated less efficiently than if that gene had evolved in bacteria, because the codons that are used in humans are actually slow to code the protein. The prokaryotes have no nucleus, so transcription and translation are simultaneous. That's something to remember, is that these two processes always go together in bacteria because there's just no nucleus. You can't separate the mRNA from the ribosome. As soon as the RNA is transcribed, it's translated because the ribosomes just grab it as soon as it's born. So microorganisms are everywhere on the planet. This is a very big difference with every other sort of organism. There is no place without bacteria or archaea. They grow under the ocean, in hot vents, in frozen lakes, and even outside the space station. That was the very first blog post on the Genomic Odyssey. It was the existence of bacteria that are on the surface of the space station. So they literally grow absolutely everywhere, even in almost outer space. So these are examples of where they've been found. So there were these lakes that were below 2.2 miles of ice, and they were opened for the first time after 2000, digging a hole through these couple kilometers of ice. And then you get water, which is below that, and it's still liquid because there's a huge amount of pressure and the temperature is not so low. Also with the salt, it can be liquid even below zero. And in there, they found bacteria that were living in conditions that are absolutely, impossible for everything else. They also found them, it was very surprising, in the 70s in Yellowstone, in this hot water that is like almost boiling, 100 degrees sometimes, and full of sulfuric acid. Some bacteria are just fine there. They're mostly archaea actually. They love it there. It's their comfort zone. You also find bacteria there under the water in the hot vents where there is no light. And so the way that they use the cycle of energy is completely different, not based on photosynthesis or input from the sun. So basically, microorganisms, you find them literally everywhere you look at. And now, of course, they're on Mars, they're on other planets because we have sent probes. And so it's impossible to decontaminate completely the instruments and the space shuttles or whatever we send there. And so it means that we have sent bacteria on Mars. Maybe they died on the way, maybe they're dead there, but maybe not, who knows. So the concept of species is kind of different in eukaryotes and in prokaryotes. In eukaryotes, it's based on sexual reproduction. If you can have babies together, then you're from the same species. But bacteria don't really make babies this way. So the definition of species does not apply to microorganisms because their reproduction is asexual. The definitions are empirical and based on homology between genomes. The species of microorganisms are called operational taxonomic units, or OTUs. And so here you've got a tree of life of bacteria. And so every time you have something that is considered to be the same species, you just say OTU when it's a bacterium. You don't really say species. It's not because they cannot have babies together where they can have babies together. It's because you compare the genomes using methods that we've seen before, okay, like pairwise comparisons, and you say if they're like 90% identical or something like that, then you'd agree they're the same species. And if they're less than 90% or whichever threshold you decide, then you'd say they're different species. But you would not use the word species, you'd say OTU, not species. Okay? So different definition of species. So the main method to study the diversity of OTUs in samples of bacteria and microorganisms is the 16S or RNA. That's the method that we've seen before. This is one type of barcode. That's the way you can identify a species by just doing a PCR on its genome. So the microbial 16S or RNA has 9 variable regions flanked by constant regions. You can see the variable regions here in gray and the constant regions are there in green. This is ideal for DNA barcodes. The most widely used region is V4, the one here between 700 and 800 more or less. It is considered that 2 sequences are from the same OTU if they are 98 to 99% identical. So that's on the 16S ribosomal RNA. I'm not talking on the genome. Okay, like this is an extremely conserved region. So when something is like only 97% identical, we say, oh, well, that's a different species. So you put your primers in that green region here and that other green region. You do an RT-PCR, and you would sequence whatever you find here in this V4 region. And then if you find 2 sequences that are kind of 94, 95% identical, you'd say I have at least 2 different OTUs, maybe even more, in my sample. Okay? So that's what we've seen so far. That's what we've seen before. Are there any questions so far with what we've seen? Yes?
So it's saying that variable 4 is like that region is like from what I understand, is like universal for all bacteria. So that's like the region we would check to see if they're similar.
The question is whether region V4 is universal for bacteria. Yes, it is. like V4 is variable, but it's flanked by two sequences that are not even variable. Like every bacterium out there, probably even every archaea, has exactly the same sequences in green on the left. So you can't put your primers there because then they will anneal perfectly, and you will see some differences in the V4 region. But this, it's universal. It's a ribosomal RNA. Every organism has ribosomal RNA, otherwise they can't translate anything. Okay? Viruses do not have ribosomal RNAs because they can steal them from the cells that they infect. But every organism that can translate needs these ribosomal RNAs, and those sequences that are here, they are universal. I mean, they're conserved enough that you can put primers. So why are these? What's the pattern here with these regions that are totally conserved interspersed with regions that are less conserved? Is because the regions that are in green here, they must be the catalytic center of the ribosome. Right? The catalysis in ribosomes is not made by the proteins, it's made by the RNA. The proteins are there just to make a scaffold for the RNA to fold in a way that is making them active. So you can't mess up with the green sequences at all, because these are really the ones that do whatever ribosomes do, which is to make proteins. The ones in gray, they can be loose between these catalytic center or something like that. So they're usually for support. So as long as they have the more or less correct structure, that's fine. But you don't need them to be exactly one way. They just need to have a more or less the right structure. So they can evolve a little bit. They evolve very slowly, of course, but they can. Yes, there's another question.
On the last slide, you said that they are identical if they're 90%, like identical, and then like on the 16S slide, you said they're not identical if they're 94 or 95%.
Yeah.
Why?
So the question is, why do I say here the identity would be 90% compared to here 98, 99%? So the reason is that when you compare genomes or when you compare ribosomal RNAs, the level of conservations are not the same. Ribosomal RNAs will evolve extremely slowly because they're really conserved. Like you cannot mess with their sequence at all. You can change it just a little bit and they will still be functional, but not too much. So the rate of evolution of ribosomes is one of the slowest evolving sequences ever. And this is why you can use it for making trees of life, because you can still recognize them from the way they were 3.5 billion years ago. This is absolutely insane. That's a time that's really unimaginable, 3.5 billion years ago. And to say they haven't changed much in 3.5 billion years is already absolutely amazing. Genomes is a different story because genomes are full of different things. Among others, there are like repeated elements, like they can have the insertion of a retro element or something like this, which already make a big difference. Like it could be plus or minus 3 kb or something like that. So that really goes beyond the 99% identity with just one event, if the species can tolerate this insertion. So genomes will have insertions, deletions, also proteins that are not that important. They're important but not that much. Or they have many ways to be functional. They're much less conserved, much less constrained, so they can evolve faster. And this is why when you compare genome, I think usually the threshold is somewhere around 85%. You consider that if the genomes are 85% identical or more, then they will be the same OTUs, and if they're less than 85% identical, then they're different OTUs. So it really has to do with the speed of evolution, but that's completely arbitrary. Okay, in a sense, the notion of species doesn't really make sense for bacteria. You should really see them as a tree without really separation between the species very much. How's that going for people listening online? Can you follow when we think this way? You can also ask your question on the chat. I'm going to read them for everybody so that everybody can see them. Yeah, they see. Yes, good. Okay, fine. So let's move on. So metagenomics, what is it? Metagenomics allows us to study microorganisms without culturing them. So most cannot be cultured anyway. Conceptually, it is just a shotgun assembly of reads from microbial communities. So before metagenomics, before we could actually sequence that much amount of DNA or whatever we want, the the only way to study a microorganism was to culture it. Like you take a petri dish, you spread whatever sample has been contaminated or contains it, and then you hope that it will grow, that you get these spots looking like that, and then when you get it, you can sequence it and see what is it, or maybe do some more tests, etc., and see what's inside. So that was the method before 2005, 2010. Culture of one targeted presumptive bacteria and characterize the genome by sequencing it or whatever that is. And so the issue with that is that we cannot culture most of the microorganisms. They grow in conditions that we cannot reproduce in the lab. So some of them, we know how to do it, but it's just super difficult. For example, when they're anoxic, when they're killed by oxygen, this is already extremely difficult to work in the lab because you must make an area in the lab that does not have oxygen. But we do need oxygen, so you need to have a separation that makes it very, very difficult to work with. And as soon as you got a little bit of oxygen going in their chamber, then they would all die. So in that case, we know what to do, but it's just very difficult. In some cases, in most cases, we don't even know what to do because we can find these bacteria in the wild, but when we try to grow them in the lab, they just refuse. One thing is missing. It could be one of really many things. Think about the pH, the temperature, any sort of nutrients or chemicals that could be in the earth that we've got no idea about. Everything goes. In metagenomics, that's not a problem because we get the shotgun approach. So we just grab the sample, extract the DNA or the RNA, whatever we like, and we sequence it. And after that, we can assemble the reads with the techniques that you've seen, using spades, for instance, and that gives us an overview of the genomes of the bacteria that were in the sample. So that's the metagenomics approach. It just means get the samples, sequence, assemble, you're done. So with these methods, we've discovered a lot about life on Earth, about the invisible life on Earth. And the first place that people started to look for them was the oceans. The oceans are full of microbes. The Terra mission travels through the oceans and samples microscopic marine life. Their first mission was to collect plankton, but they still managed to assemble approximately 2,600 prokaryotic genomes from their samples. So for a comparison, we knew at the time approximately 30,000 prokaryotic genomes. So in just one trip, they increased by 10% the amount of known bacteria. And without even looking for them, they were looking for plankton. Okay. And since 2020, the new Terra mission is to sample the microbiome specifically. So they have this boat here, goes around the globe. So these are the places where they've been already. So they just keep going around. And then they send a probe down a different depth of the water. And they just collect a bit of water, bring it to the surface, and on the surface there, they have a molecular biology lab and sequencers, so they can just get the DNA directly and sequence them on the boat. So they do this a little bit randomly, going to different places, sampling water at different depth, and now they're kind of looking specifically for prokaryotes. I don't know what they have discovered, but like this kind of mission keeps discovering more microorganisms that we never heard about. And these are things that we could not grow them in the lab. What you need is ocean water at a certain depth, a certain temperature, a certain amount of things in it that we can't really reproduce. Sometimes even like the wave or the current are actually quite important for the growth. So all we know about these organisms is where they were sampled and what their genomes are. And you'll see sometimes that's enough to know a little bit about the biology of these organisms. For instance, archaea contribute to the nitrogen cycle on this planet. Ammonia oxidizing archaea, also abbreviated AOA, grow at depth below 2,000 meters and at temperatures of 4 to 10 degrees. All we know about this process comes from metatranscriptomic analyses of deep sea water to reconstruct the metabolic pathway. So what does that mean? Here you've got a map of the world looking more or less like that, and people have sent, again, like different probes, going like really deep. We're talking 2,000 meters deep, so these are pressures that are kind of unsustainable for us or for most of the animals, basically. But you can still send probe, collect the water, and look specifically for one particular organism, and it could be archaea. So they grow at relatively low temperature, we're talking 4 to 10 degrees. But using metagenomics, it doesn't matter very much. You just collect it all, sequence, assemble, and then you get the genome. And once you get the genome, you get the enzymes. So by homology, you could say, oh, this enzyme has got this domain, it's been found in E. coli, and so it just does this in E. coli, so you must still do the same in this organism. So most of the time they can say, okay, we've got these 10, 100 enzymes, and these enzymes, we can actually synthesize them in the lab if we need, and we can study what they do at the biochemical level in the lab. We don't need to grow the organisms for that. We can just produce their proteins in vitro. And with that, you can have an idea of the metabolic pathways of what they're doing inside the bacteria, but also outside the bacteria. Because you find all these enzymes that are present within, I don't know, 1 liter of water, whatever that is. And you could say, okay, these metabolites are going to be produced in this bacteria. They're going to then transfer. There's going to be another bacterium that's going to metabolize them in a different way. So you can construct metabolic pathways that look like that. So these are a little bit like the Krebs cycle that you must know, or this kind of metabolic pathway where one particular metabolite goes from one state to the other, and the enzymes that are present are going to catalyze the transformation from one into the other. So you can see what metabolites are produced and how they go through this metabolic pathway to produce something else. And because there's just so many of these archaea, they actually influence the whole nitrogen cycle on the planet. This is really something that is totally invisible for us. It's not even happening in the atmosphere. It's not even happening at the plant level. That is kind of much easier for us to study. It's happening way in the deep at 2,000 meters, where we cannot send someone to do experiments. It's just way too difficult. The only thing we can do so far is to collect the DNA, sequence it, and see what's probably happening there, and reconstruct these metabolic pathways. And that tells us something about the ecology on the planet as a whole. Right, is that clear, Desali? Yes.
I was wondering, is this method the same as collecting eDNA?
Is this method the same as collecting eDNA? It almost. Collecting eDNA, you like grab the DNA, but you would do a targeted PCR. You remember that you've got these barcodes, so there were these genes that we mentioned, like RBCL, etc. So you're just looking for the identification of the species with eDNA and DNA barcodes. In a sense, okay, you could say that bacteria are almost an eDNA. It's like they don't release their DNA in the wild, but you can just catch it there. So it's really almost the same. But in general, eDNA, environmental DNA, just means that a part of the organism that has left some of its DNA. It's not applicable for bacteria, but the idea is very, very similar.
Would you say metagenomics is the best way to study like microorganisms now, or can you improve it at all?
So is metagenomics the best way to study microorganisms now? It's not the best. It's the only one for the microorganisms that we cannot culture, which is 99.9% of them, basically. Right, so we don't have an alternative.
Will there possibly be different alternatives?
Will there possibly be alternatives in the future? Yeah, of course. We can imagine that we would be able to culture them. Maybe one day, maybe even by looking at their genome, you could say, oh, well, obviously this microorganism needs that because it doesn't have this enzyme, so it needs to find it already in the environment. Maybe one day we'll make so much progress in terms of being able to understand the relationship between genomes and environments that we'll be able to tell what we need to culture them. If we can culture them, then yeah, that's kind of nice because you can do much more than just looking at the genes. You can also look at, interspecies interactions, other things that are pretty difficult with metagenomics. So it's not really that it's the best, it's that it's the only one at the moment. And will there be better ones? Yeah, I think so. I think with the advancing technologies and the advancing understanding of the genomes, I think at some point it's going to be better. All right. So what about viruses? You can also do the same for viruses. You can do it obviously for bacteria, for archaea, but what about viruses? Viruses is a little bit more complicated, but we can do that at the moment. shotgun assembly is useful to identify new viruses and new viral genes. First, we assemble contigs, then we search virus-only genes. From this, we know that the genes on the same contig also belong to viruses. Okay, contig, remember, you take the reads, you just pad them, you don't stitch them next to each other. We've done that during the midterm. So piece of cake now. So you get contigs. And within these contigs, you can look for example, capsid genes. If you find a capsid, you know you've got a virus. Like capsids are really something that exists only for viruses. So you look at the contig, and if here is a capsid on the left, then whatever is on the right, because it's on the same contig, it's on the genome of the virus. Therefore, it must also be viral genes. Okay? So you've discovered new viral genes with this method. Like again, use assembly as a way to discover new things. The difficulty with that is that some viruses are integrated in the genome. For example, the lambda phage, if you're familiar with it, can live its life outside, can also go inside the genome and then integrate. So you'd get a long contig, and you'd see the capsid somewhere, but you also see like 4 million other nucleotides in the same contig. So you don't really know which are the genes in there that belong to a virus versus the one that belong to a bacteria. So this method is not 100% sure, but at least it gives you a start. And in terms of viruses, the problem is that we so much don't know what to look for that we don't really know the diversity of viruses. The genomes are very small. Sometimes they're not well assembled on top of it, so that makes smaller contigs of size 100, 200. Everybody throws that away, say there's nothing interesting there. So we don't really know what to look for, and unsurprisingly, we don't find it because we don't know what it is. But that's one way in which people have been able to discover new viruses in different samples. So with that, one problem, of course, is that you've got the virus, but you don't know what's the target, what does it infect? And a pretty cool trick that has appeared over the years is to use CRISPR for that. So CRISPR, you may be familiar with it. If you've watched any science fiction TV series, you probably have heard about CRISPR. Probably you haven't heard the right thing if you've done just that, but you've heard about it. It's now completely mainstream. So that's a technique to modify genes, that's a technique of biotechnology, but originally that was something else. CRISPR revolutionized biotechnology, but it is originally a defense mechanism against viruses. That's the immune system of bacteria, CRISPR. CRISPR arrays can be used to know which phage can infect which bacteria. So how does that work? Here you got a CRISPR array. So it stands for clustered, regularly interspersed palindromic repeats, something like that. So it just means it's a locus in the genome where you have these red regions that are always the same and the green regions are different. And the green regions, they're actually a sequence that comes from a virus, and with the protein Cas9, the bacterium is able to get that sequence in an RNA, it's transcribed, load it onto Cas9, and when the DNA of the virus comes inside the cell, there will be a match between that particular sequence and the genome of the virus, and it's going to trigger the cut of the virus. So this is a nuclease, Cas9, and then the nuclease that's directed to cutting incoming viruses in the genomes of bacteria. And it's been repurposed to cut whatever we want, because here these sequences that are in green, people have realized, well, I can just choose my favorite sequence in the human genome or in the pig genome or whatever that is, and then I will be able to direct Cas9 to make one cut in the genome instead of cutting a virus. So that's how this was repurposed. So when you assemble a new virus and then you find one piece of that virus in the CRISPR array of a bacterium, you know one thing, is that this virus can infect the bacterium. This is the only way this bacterium can have put this piece of DNA in its CRISPR arrays. The bacteria, they don't have like B cells and T cells. Their memory is that they steal these pieces of the virus and they put them in the CRISPR locus. If they have it there, it's because they have been infected before by this virus, so this virus can infect them. So you can match the CRISPR arrays of bacteria with the viruses that you sequence using these methods, and that tells you there's a match between these two, so this virus must be able to infect that bacteria. So you can also do viral metatranscriptomics. Okay, we're kind of mixing up everything that we've seen so far. Many viruses are in RNA form all the time, and all of them need to produce some RNA. Even if they're DNA, like HIV, for instance, they have genes, they need to transcribe them, so they all have an RNA form. So you can just take RNA, do reverse transcription, and then you go metagenomics, and you see what you find. And when you do this, you can discover plenty of new viruses. I'm going to show a particular study here, where they started to be interested in viruses of invertebrates. We know most of the viruses of vertebrates, at least we think, especially of humans. But until recently, we had no idea whatsoever whether other organisms have viruses. Are we the only ones that have viruses? Or are viruses very frequent? Or they're very scarce out there? How does that work? So people just started to use this method, like get a couple of insects, crustacean, other kind of organisms, doesn't matter what, just grab them all and do an RNA-seq on them to see what kind of viruses you're going to find. Invertebrates are full of viruses, like every other organism, actually. Metagenomics reveals on average 5 to 6 viruses per sample. Every organism that is an invertebrate has 5 to 6 viruses that you can find relatively easily in their sample. Adding up to potentially large fraction of the mRNAs. Most of the invertebrate RNA viruses were identified in one single study in 2016. Okay, so 95% of what we know about these RNA viruses comes from just one metaviromic study. It's this one here. When you look at some of the information on the y-axis, this is actually quite scary. This is the proportion of RNA coming from that sample that is attributed to viruses. And you can see that these poor insects here, it can be up to 87% of the RNA are produced by viruses. So most of the RNA transcriptome was actually taken over by viruses. I wouldn't like to be from the species of these guys. For other groups, the numbers were a bit smaller. In humans, it would be much smaller, of course. Like most of the time, we're not infected by viruses that take over completely our transcriptome. But in invertebrates, it seems that the levels are much higher. And one of the questions is, how do they tolerate that? You know, how can they even live with something that steals 90% of their RNA production capacity? Maybe this particular sample was actually dying, we don't know. But they observed overall a very high level of infection, not just infection, but also the burden of parasites, of viruses in these species was much higher than in vertebrates. And how is that possible? How can this be sustainable for insects, especially that their immune system is really not at all like ours? It's a bit of a mystery, maybe a question for you guys to answer. So the newly defined Dombus noda clade of RNA viruses is one of many examples where segmentation, the number of chromosomes, keeps changing. So in that particular study, because they had sequenced really 95% of everything that was known in invertebrate viruses, they decided, let's make the new classification, because the previous classification did not make sense. In just one study, they could do it all. And before this study, they thought that segmentation was really a big thing. Like if the virus has got 2 chromosomes, or if it's got 1 chromosome, then they're going to be very different distal clades. Because, come on, like going from 1 chromosome to 2 chromosomes is a big event for evolution. Turned out it was completely wrong. So now that they had a much bigger spectrum of the viruses that were existing in these species, they could see that viruses always change the number of chromosomes like it's nothing. It's not like vertebrates or mammals where acquiring 1 chromosome means a new species or losing one means a new species. In viruses, that happens all the time. Before this study, segmentation was used for classification. It is now considered uninformative and shows how fast viruses evolve. Okay, so with these metagenomic studies, just one metagenomic study, you change completely the knowledge of what was known about viruses in invertebrates. That's a little bit where we are. So when every study changes everything you know, it's because you don't know much. That's the point that I wanted to make. That's it. That's time for the break. Are there any questions before we go for a break? Yes, in the chat. They are scanning the RNA based on the viruses that are discovered to date. So if a virus is still unknown, this means that the proportion might be higher, right? Yeah, exactly my thought. Like, how can you discover something if you've got really no idea what it looks like? Imagine that a virus would be non-homologous to anything we know. How would you even know it's a virus? You just see a piece of DNA and you say, well, there's a piece of DNA. Maybe it's a plasmid, maybe it's a virus. And that's absolutely a good point. The only counter argument that I'm aware of to say that no, like this is unlikely to happen, is capsids. I think there are only 9 or 10 families of capsids that were ever found so far, and people consider that that's it. It's gonna be that, because when you think about it, the way you can make a capsid has got some geometrical constraints. You know, there's so many volumes you can make in 3D, and so the proteins must have a certain shape. They must be able to make a couple of angles that will make the like the spherical-like, or whichever shape is the capsid. And so it's considered that evolving a new type of capsid is just way too hard, and so we probably know most of them. And if we're missing some capsid, we're maybe missing one family, but not like 100. That said, that's a hunch. You know, before you know, you don't know, and so it could be that actually, at some point, someone clever is going to realize that some of these proteins are actually capsid, right? Or that the way of forming capsids is more than we thought. Maybe you can have dimers, or maybe even, who knows, one part of the protein coming from the host, one part of the protein coming from the virus. And so how can we know that? Probably experimentation. You need to produce the proteins in vitro, see whether they're able to form special structures. So at some point, when we can not only do massive sequencing, but also massive production of proteins, we could discover some new properties of these proteins. But so far, the consensus in the field is that there won't be a completely new type of capsid, which means that there won't be a completely new type of virus. I hope that answered the question. Other questions? Yes?
I think for slide 9, we talked about the 16S RNA, and then you said we use the region for DNA barcode. Is it the variable region 4, or is it the constant region that's like between 3 and 4?
Yeah, so the question is whether V4 here is the barcode, or is it the region between 3 and 4. You can use any of the 9 regions for a barcode, from V1 to V9. So you have 9 potential barcodes, or you can sequence the whole thing, and that gives you one bigger barcode. Okay. So why are the green regions not really good for barcodes? It's because they're exactly the same no matter what species you look at. So that'd not be a very good choice for a barcode. Now a question for you: why not use the whole 16S RNA? Actually, you know, like, you get, of course, more information by looking at the whole RNA than looking just at region 4. So are these guys lazy, stupid, or what?
We would make the cost of barcoding higher.
Okay, the cost. It could be the cost. So here we have something that's around 100, maybe 200 nucleotides. It would be more expensive to sequence something that's longer. That's definitely one of the reasons. The other one is just the feasibility, as simple as that. Remember your classes on high throughput sequencing. Most of these sequences are made with Illumina, and Illumina has got a hard threshold at 2 times 250. Okay, so already the whole 16S is too big for Illumina. And if you used methods like nanopore, what would be the problem? Higher rate, of course, higher rate, because nanopore, remember, is like 10% differences. So if you're looking for identity of 98 to 99%, you cannot, right? Because you'd get more errors added just by the sequencer. So you'd keep finding new species. So you need to be something that is more accurate than 98 to 99%, because that's the threshold. And nanopore is just not there yet. The technology is increasing. Like, it's probably around 5% now, maybe 3% even. But that's still too high. When you cannot tolerate more than 1 or 2% errors, then you have only one option. It's Illumina, which means that you cannot do the whole 16S RNA. Okay. All right. So we're going to make a little break now. Let's say 5 minutes. We meet again at 3:05, and that's going to be Kyle taking over. Okay, we're back on the recording. So now we're going to welcome Kyle, who's going to give us a short introduction about how to get started with your blog post. So, Kyle, go ahead.
Okay. Hello, everyone. My name is Kyle, and I'm here to give some blog post writing tips that may be helpful to you as you continue constructing your blog posts. Oh, what's
Just click on the screen.
the screen? Oh, okay. Oh, this? Okay. All right, just to start, just a brief introduction to myself. Again, my name is Kyle Valentino, and I'm currently in my 4th year here at UTSC, specializing in molecular biology and biotechnology. I took BIOD25 last winter term with Dr. Filion, and I really enjoyed my experiences in the course. So essentially, I want to reciprocate that experience and pay it forward by suggesting some advice that may be helpful to you. And so my advice mainly surrounds 2 ideas, which is to start early and ask questions, where starting early will allow you to maximize your time. And asking questions will be you taking advantage of the wealth of resources around you, whether that be Dr. Filion, your TA, or each other. I'd also like to take this opportunity to open myself up to you guys as a resource, whether that be to bounce off ideas, ask questions, or anything of the sort. You can find my email on the slides. And so, just to show you guys how I applied my 2 pieces of advice, I prepared a little anecdote for you. And it goes like this. Once I started writing my blog post, which was at a sufficient time before the due date, I figured out that I could use my past courses to my advantage. Specifically, I wrote my blog post on a GWAS, a genome-wide association study, on Alzheimer's disease, leading me to review a past lecture from BioC14 titled Genes, Behavior, and the Environment, as well as lecture 9 of this course, BIOD25, which covered GWAS, for essentially background information on Alzheimer's disease and the GWAS, which I in turn applied in my post. I also did this same thing, i.e. referring to other courses in other areas of my post. And I encourage you to ask me, where else? And so, oh no, I have one more part. All right, so yeah, just the main idea. So yeah, just to bring everything together, the main idea of my anecdote is that once you start writing your blog post, understand what the expectations are and think about what could help you to essentially use your time to compliment yourself and to strive to make the blog post a team effort through, again, asking questions and utilizing your resources. And one final piece of advice I can give you is that some of the blog posts from last year are available to you on the Genomic Odyssey. So I say, take advantage. And with that, that concludes my mini presentation. Thank you very much.
You don't want to go through the other slides?
Okay, I can go through the last one.
Is that important? Or
Yeah. Kind of wanted them to ask me a question.
Oh, sure.
Hold on. But are there any questions for me right now?
So
Sorry?
where did you use?
Where else did I apply other courses? Okay, great question. So bear with me for a second as I go through this bonus slide I prepared for you guys. Essentially, this slide outlines how I applied the inverted pyramid template on the far side that you guys will be using to write your blog posts. I'll go through it. And so it goes as follows. First, the most newsworthy information, important details, and other general information and background information. And now in regard to where else did I apply other courses within my post, in the 3rd section, other general information and background information, I wanted to give readers an idea of the pipeline associated with research in general, i.e. going from a petri dish at the bench to actually applying therapies at the bedside. So, and recall my context is Alzheimer's disease. So, when it came to therapies, I recalled or thought about different therapies that were taught to me throughout my undergrad. And one that was apparent to me was monoclonal antibodies. Maybe I'll highlight it right here, down there, which was highly talked about in courses such as BioC39, immunology, and BioC23, biochemistry laboratory. So yeah, I did the relevant research. Once I recalled monoclonal antibodies, found out there was a monoclonal antibody available for Alzheimer's disease, and put that in my post. Does that make sense? Any other questions? Yes.
So, just out of curiosity, like, when you were writing your blog post, like, how did you do it? You said you started early. Like, did you do it in chunks? Like, you literally spread it out?
Yes. Me personally.
Repeat the question.
Oh yes. The question was, how did I write my blog post? Did I do it in chunks, or essentially how I went through things? It's a pretty tough question. Me personally, I read my paper in chunks, so the methods, the abstract, etc. And I gave like a day for each. And then for the different sections, yes, the 3 different sections, I spent a day on each section. Is that clear? Wait, yes.
What distinguishes like your blog post from the actual article itself? Like what makes it stand out? Is it the way it's written? Because I know the article sometimes will cover the scope of the topic.
Yes.
Like what makes your blog post, I guess, or what would make my blog post different?
So the question was, what made my blog post different, and what could you do to make yours different? Well, I guess for starters, I included other general information, background information, where the stuff I included did not necessarily, or was not necessarily covered in the paper. For example, monoclonal antibodies. But yes, for the most part, the first 2 parts, the most newsworthy information, the important details, will be essentially a reiteration of the paper. There. Any other questions? Yes.
Did you find it better to make your own figures, or was it better for you to use the figures from
So the question was it better to use my own figures or figures from the article? A mix of both. Depends on what figure you're using and what you're trying to explain. Essentially your message. Yes.
So, would you say when you're writing the, like you read the blog, you read the article,
Yes.
and then you're supposed to basically talk about it. So how do you know which parts are more important and which parts are less important to write about?
Yes. So the question was, what parts of the article are more important or most important? I think this will be covered next week when Dr. Fumio goes over how to read an article. But I guess I would say, look for like in the abstract to start. Look for like action words. For instance, we found our findings uncovered, and then build from there. That make sense? Are there questions? I actually have no idea. Apologies.
We also need to wrap up. So, what was the hardest part about the writing process for you? How did you avoid procrastinating?
I think everyone heard that. I guess I started early, just intuitively. Yeah, started early. I knew I had to start early or there would be consequences later on. So I put that in my mind. Start early. Yeah, and yes.
Was it important to use BIOD25 topics, contents, or can you search that info on the internet?
So the question was it important to use BIOD25 content in my paper? I think for everyone this varies. Specifically, I did a GWAS, which it was covered in BIOD25. So it really depends on your paper and what it covers.
Now tell them that they have to do anything else than BIOD25. They gotta get a new thing.
I'm sorry?
Tell them to get new things, not just content of the past. No, who cares about BIOD25? Is it like writing a review or a reflection article?
Interesting. okay, so the question was, is it like writing a review or a reflection article? I see a bit of both. Maybe at the top, it'll be a review. At the bottom, you have a little more freedom, like I showed at the bottom. So there might be a reflection or connecting to other concepts.
I'm gonna mix up both. Okay, so let's wrap up now because we need to make progress for the rest.
Thank you very much.
Thank you very much.
Okay.
Oh, gee.
Okay, thank you very much, Kyle. I really appreciate that Kyle did this. Also because, of course, when I tell you to start early, you won't believe me. But when it's a student, now maybe you're going to think about it a little bit. I'll also give other answers to the questions that were asked. Like, is it like writing a review or other type of article? No, it's a blog post. It's really a blog post. I mean it. No kidding. It's a blog post, which means it's got to be fun. It's got to be interesting to read. That's the most important. If you've got no reader, you did a bad job. If you've got plenty of readers, then that's an amazing job. The hard part of the exercise is to take something that's horrible and complicated to make it simple and fun. And that's horribly difficult to do that. But if you're able to do this, it really means that you're able to process complicated information. And that's what I care about. If you can make it fun and understandable, it's because you understand it. You can't be fun if you don't understand what you're talking about. You can't be entertaining, right? So it's really about making something that's inaccessible and, like hard to access, to make it accessible for people, basically for you, for other students. The target audience is the students of next year, before they take BIOD25. So, any questions you'd have, you can ask me, but even better, you can ask Cal, because guess what? He's really social. He likes to answer your questions. He's really motivated. He's still, on the blog. He's writing comments on the post. You can see him. He's everywhere. Thank you, Cal. So let's go back to now metagenomics, and the second half is about human health. Almost all the sicknesses affect the gut microbiome, even mental illnesses. So do half of the most common drugs, and of course, so does diet. We can thus use bacteria as biomarkers and diagnostic tools. So that's an idea that's actually very, very deep, and that will have huge consequences for the medicine of tomorrow, is the fact that because whatever we look at, every disease, including mental diseases, no kidding, mental diseases change the bacteria that are in your intestines. So it means that if you just know the bacteria that are in the intestine of someone, you kind of have an idea of what's wrong with them. And maybe you have an idea of how you can make them better. And that's a completely new idea for medicine that did not exist 10 years ago. So in order to work with microbiomes, we need a couple of tools. And one thing that we need is a way to measure diversity. So there are two types of diversity that are called alpha and beta. And the alpha diversity, the one that's here, is the average variability within a community. Okay, so think about it as Scarborough diversity. We are inside the campus, inside this part of town. We are very diverse. There is no majority in Scarborough. Everybody's a minority. As the alpha diversity is the Scarborough diversity. There's also the beta diversity, which is the comparison between two samples or two sites. It is the average variability between communities. So you can compare, for example, the place where everybody is identical. Let's say they would be all, white males. Another place where they're all black females. They have a huge beta diversity because they don't look anything like each other, these two places. Okay, so even if each of them is individually not very diverse, between them you have a huge diversity. So that's the beta diversity. So that'd be comparing where you are to another place, or comparing the microbiome of one person to the microbiome of another person. So the one that we care about the most for health is the alpha diversity. Within the person, what is the balance or the imbalance between bacteria? So metabolic pathways, what we've seen with the archaea living in the water, we can also do the same for the bacteria living in the guts, or the archaea living in the guts actually, because of course There are a couple of archaea, they're not all bacteria. From assembled genomes, it is possible to extract enzymes and reconstruct metabolic pathways. The healthy gut microbiome is different between people, but the metabolic pathways are more or less constant because different bacteria have the same enzymes. So this was the big discovery of the very first microbiome studies, is that you look at the microbiome of two different people, they're very different. But when you look at their metabolic pathways, they're the same. Right? It doesn't matter which bacterium comes and brings you this particular enzyme or this particular byproduct. As long as this enzyme is there, that's all your body cares about and you're healthy. Okay, so you have plenty of options for being healthy in terms of microbiome, and these options are kind of explored a lot by the population. The microbiomes of people are different, but in terms of what these microbiomes do, in terms of enzymes, it's actually quite the same. It's quite constant. Okay, that's what this means here. Again, you have here, for example, the tryptophan metabolism pathway. And so one thing that is important to remember is that we have what? What's the number of genes in the human genome again? Order of magnitude. 2,000? 200,000? 20,000. Okay, we have 20,000 genes. Many of them are not enzymes. We say we have approximately 5,000 to 10,000 enzymes, probably closer to 5,000. And bacteria have got tiny genomes, etc. But there's just so many of them and so many different bacteria that we get somewhere around 50,000 enzymes from these guys. So much more genes coming from not our organisms inside our body. So we expand our metabolic capacity quite a lot with this microbiome. You can see this a little bit as evolutionary farming. Our body has learned to kind of farm these microorganisms because we need their metabolic capacities. We eat grass because, well, we don't eat grass, we eat plants because they can metabolize the sun and we cannot. And so it's the same. We kind of use bacteria because they can do metabolic operations that we cannot. And in exchange, we allow them to live inside our guts. So a lot of the metabolism happening in an individual, in a healthy individual, is actually coming from the enzymes produced by bacteria. It's really important to understand that. It doesn't matter which bacteria produce them, as long as these enzymes are here, you're healthy. Another question, which is quite interesting, and I didn't know the answer before I started to teach it, is the microbiome genetic? Like, we don't have the same microbiome, but is it because we don't have the same genes? Like, are twins, identical twins, do they have exactly the same microbiome or not? Think about it for a second. Make a guess. I'm not going to ask you, but just pick your side. Okay. Twin studies showed that parts of the microbiome depend on the genotype of the host. So to some extent, yeah, you take twins, they're more likely to have the same microbiome than two non-twins. Okay. On the left is the GWAS that we just mentioned, the genome-wide association study. We're going to see in a couple of weeks a lot more about this, but I wanted to introduce the concept now for the presence of this bacterium, bifidobacterium, showing that the LCT allele, that's a lactase, has an influence. Okay, so you've got the 20,000 genes here on the x-axis. The y-axis is something that we're going to explain a little bit later, which is a significance test, but in a nutshell, the higher it is, the more there's an association between the particular allele of the gene and the particular phenotype you're looking at. And the phenotype you're looking at is whether this bacterium is present, bifidobacterium. And so if you have one allele of the LCT gene, then you're more likely to have this bacterium. And there we can understand it. Okay? Either your body is able to metabolize one particular product that will create some nutrients that some bacteria can eat, some others cannot eat. So that makes sense. And sometimes you have these associations. That allele, the bacteria can grow. You don't have the allele, the bacteria don't stand a chance. They don't have what they need to grow. But mostly it's not like that. These are actually extremely rare. A few more associations were found, but overall, the gut microbiome is not determined by the genome of the host. Change diet, you're going to change your microbiome much more than your genes. Okay, like two twins that don't eat the same thing, their microbiome would be mostly very, very dissimilar. Okay, it changes over time and depends on the lifestyle. So even within your lifetime, it's not the same. Okay, like of course as a baby, when your metabolism is completely different, that's understandable. But even as an adult, you can consider that every 5 or 10 years, complete flush, complete reshaping of your microbiome. Of course, you don't feel it, But if you did your microbiome analysis, you'd say, oh, well, that was me 10 years ago, that's me now, it's got nothing to do. So the microbiome is mostly non-genetic, even though there are cases where some alleles influence the presence of some bacteria, but not the whole microbiome. Right, so for Seguin, any questions on these concepts? Nothing in the chat. So there are some things that are a little bit famous, like the effect of antibiotics. Antibiotics lower the alpha diversity of the gut. In the time course on the left, where time goes from top to bottom, the first antibiotic treatment creates a succession of two monospecies with B. ceftriaxensis and then P. distosonis. So you have here the colors represent the different types of bacteria. Time goes like this, and so there was an antibiotics treatment here, and you can see that with beautiful diversity until day 60 for that person, and at day somewhere between day 60 and 374, there's only one bacterium that takes over the whole thing. Like the gut become completely dominated by just one species, one OTU of bacteria. Okay, happened again here at that point. And after this, the microbiome stabilized again. But look at the time in days. We're talking of something like 3 to 4 years. Okay, so like the antibiotics treatment has a very, very profound effect on alpha diversity, but that's obvious. Some bacteria survive, most die, and the ones that survive, they take over because they can grow and the others cannot. Did you have a question?
Yeah, how do you read the graph?
How do you read the graph? It's a stacked bar graph, except that it's been smoothed. So time goes like this, and you've got every color represents a different type of bacterium here. So green means I don't know, unclassified, and the one that are like in a pinkish color here, that'd be this species here, Distosonis, and you could see that Distosonis completely takes over here at day 377. Almost everything is that color. So that's what it means by a monospecies event. Like everything else has been pushed to the side, and Distosonis won the Game of Thrones.
But it decreases, right?
After that, it decreases. Yeah, like here it's super strong, and then it comes back to, let's say, more or less where it was before, 4 years before. Won't the microbiome evolve resistance if someone constantly take antibiotics? Indeed. So there will be there will start to appear resistance to the antibiotics, but not every bacterium will be resistant. Some will just never recover. That was like total disaster. They will just not recover at all. And remember that the microbiome changes anyway. Some species come, then it's ecology. At some point, a stronger, better, more adapted species comes and takes over, and that's it. That's life, the circle of life. And so, if the person is constantly under antibiotics, yeah, for several years you can expect that it will be only antibiotics resistant bacteria in the gut at some point, but some will never acquire this resistance. So it's not a very good idea to take antibiotics for the rest of your life. Low alpha diversity is bad for health because the metabolic pathways are perturbed. Okay, now you get the concept. Is that it's not a good idea. You need these enzymes for your health. You don't have them in your body. So with antibiotics, yeah, sure, you remove the bad guys, but you also remove the good guys. And the good guys have got enzymes that you need. So if for several years these enzymes are not present, then whatever you eat, it doesn't matter. You're not going to produce these metabolites that are important for your health. So antibiotics treatment is a great idea when your life is in danger, but you need to restrict it in time. You have to replenish a normal flora, gut microflora, to have these enzymes. You need them for health. Because the niche is easier to colonize for opportunistic pathogens like Clostridium difficile. That's also another thing. Like if you have too much antibiotics, some bacteria are pretty nasty and they're resistant. Like this is a common cause of complications after antibiotics treatment. And so that causes all sorts of horrible stomach pain. It's not really stomach, it's in the intestines, of course, when you have that infection here, and it's hard to get rid of. So that's consequences of perturbing the equilibrium that's in the gut when you have antibiotics. Of course, antibiotics are good. They save life. Like the life expectancy has increased by 30 years after antibiotics. This is by far the most profound revolution in medicine. just second would come the vaccine, but antibiotics has been completely amazing. So I'm not saying that they're bad. I'm just saying that you have to use them wisely. So there are different types of antibiotics, and most of the antibiotics need to be inside the cell. Yes, there is a question.
I was just wondering, since like antibiotics like include diversity of microorganisms, so wouldn't we have if we have like microbiome transplantation from like family members, would that help as well?
Yeah, the question is, after antibiotics, does microbiome transplantation help? The answer is yes, it does actually. That's one of the most efficient treatment. And so if you can get the microbiome from a family member to be implanted in your guts, that will really help to restore a healthy microbiome. You have to think about it like ecology, okay? Like this is really, there's kind of a struggle between bacteria, and so there's a certain rate in which they can come inside your intestine. If you accelerate that rate by getting a transplant, then you accelerate the rate that they can recolonize the intestine. That's for anybody who's comfortable with the treatment, then it's the most efficient. Some people are not comfortable with the treatment.
Can I ask might be a dumb question, but like,
Ask away.
is it possible to live with no bacteria in your gut?
Aha, is it possible to have no bacteria whatsoever in your gut? It's not a dumb question, it's a fantastic question. What do you guys think? So if you wanted to go to no bacteria, what would you have to do? Pretty extreme measures. And so for humans, I'm not aware of any possibility of doing that. But it's been done for mice, because sometimes you need to compare the effect of just having bacteria for negative controls. And so there are these special types of mice that are bred without bacteria. But to have them without bacteria is actually very, very difficult. And of course, any contamination in the air, then you start to have bacteria in their guts. So they're extremely expensive to maintain. And so that's really what allows to make a comparison between what's the state of health without any bacteria. It's bad. You don't want to be these mice, let me tell you. So let's say I'm not aware of any human having gone through that process. It's like we're talking about technology. You have to wash the ovocytes to decontaminate them, etc. So that's nothing that we could do for humans, but for mouse it's been done, and they're not healthy. Might lead to nutrient deficiency. Exactly the point. Okay, like they're missing a lot of stuff, like their immune system, or like everything is bad. Basically, everything you look at, we need these nutrients for our health. And thanks God, it's not really possible for us to not have them. But maybe based on these experiments, the answer is we need these bacteria. Yes.
Just like another question, like my bone, like if we're like in our gut, like if it's only reduced to one like type of microbacteria or like two, is it possible for us to like recover just by like normal diet without any supplement?
Yeah. So the question is, if you have a monospecies like we've seen in the case before, can you recover by having normal diet without having any supplement? Yeah. At some point, it becomes it stabilizes. Okay, like the antibiotics pushes away the equilibrium to one extreme, but when you eat, you contaminate everything with whatever is on your finger, whatever is out there. At some point, these bacteria make their way inside your gut, slowly or fast. When you take supplements, then you accelerate the recolonization with microbiome transplant. But even if you don't, remember bacteria are literally everywhere. There's no way to get rid of them. So they will come back at some point, but you want this to happen fast. So antibiotics, how do they work? It's not really strictly related to genomics, but more and more there are some connections between antibiotics and genomics. I wanted to touch upon a little bit the way antibiotics work. Antibiotics target machineries that are present but different in eukaryotes. Most antibiotics target the ribosome, so they need to be imported in the cell. Few antibiotics work from the outside. So these antibiotics, they're molecules that really, really annoy prokaryotes but leave eukaryotes alone. That's why you can give them to cows, to humans, something like this. They do nothing to us, but they really annoy microbes. How does that work? For example, the ribosomes, they exist in us, in humans, in animals, but they're different enough from the ribosomes of bacteria that you can make small molecules that will really inhibit all the ribosomes of bacteria, but do nothing to our ribosomes, because our ribosomes are different enough. But all these things, for them to work, they need to be inside the bacterial cell. That's the problem. Like here, you've got different classes of antibiotics, and the ones that target the ribosome are here, and that's most of them. Actually, approximately 60 or 70% of the antibiotics are in this class. So the easy way for the bacterium to not suffer them is just to export them outside the cell. So this is called the multi-drug resistance problem, is that as soon as the bacteria acquire a pump that just throws out of the cell everything that doesn't look nice, then they resist to literally everything. So they have only one gene that they acquire, and then they resist everything. You have only these antibiotics that are here that can act on the outside. These are the ones that we keep really in a safe place for the last moment, because we don't want bacteria to have resistance for them. Because these ones, if they resist that, then they resist everything. Okay, they can easily acquire these pumps that give them multi-drug resistance. So the beta-lactam, unfortunately, this is too late. The resistance is just so widespread because that's penicillin here and the equivalent. But you got vancomycin and the polymyxins, for which there are resistance out there already. Like there is resistance for everything. There are already bugs that are resistant to every known antibiotics. Thanks God, there's not too many of them. But we're keeping these ones, really we're using them with extreme caution because we don't want to accelerate the resistance to these ones. And you understand the mechanism, right? Whatever needs to be inside the cell to kill them, they just have to not get it inside the cell, and they can survive. So antibiotics resistance, and this is where genomics meets medicine and antibiotics, is that in the end this is about gene traffic. You can be resistant to antibiotic only if you acquire the gene to be resistant. And how does that work? Many antibiotics resistance gene are on mobile elements, and everything seems finally to link together with what we've seen before, such as transposons and plasmids. They can be transferred by conjugation, that is DNA transfer through a pilus. I'm going to explain that in a second. Transformation, direct pickup from the environment, or transduction, like phages delivering the wrong cargo. So conjugation here, it's this case that is there. Bacteria have a parasexuality system, so they can build a bridge between two bacteria, like it's a pipe, in which they just funnel the plasmid, goes to the next cell. So like here, I'm just giving you that, do whatever you want with it. And that was an antibiotics resistant gene, for example, ABR here, okay? That's a pilus, like this thing that bridges the two bacteria is called a pilus. And that is conjugation. Transformation is that these are processes that are not fully understood, where bacteria just pick up, they eat the DNA from the outside, say, oh, that's interesting, maybe I'm going to try and express that. Bad luck, there was a virus, I'm dead. Oh, good luck, there was an antibiotics resistance gene, and maybe I survived. So they kind of bet a little bit on trying different things. And so you've got these natural transformation processes happening in bacteria. And transduction, which may actually be the most frequent, it's very hard to measure, is the fact that phages sometimes mess up very badly. Instead of making a copy of their own DNA, they take a piece of the bacterium that they're from, and that's what they package in the capsid. So what they're going to deliver to the next bacterium is not their own genetic material, it's the genetic material from their previous host. And that can be an antibiotics resistance gene. And you say, come on, Guillaume, what's the chance? Like, you know that it'd be exactly the antibiotics resistant gene, and it would be exactly that phage messing up exactly that time. Yeah, but they do it so often and they're so sloppy that in the end, that does happen, and quite fast, actually. So phages are kind of, a little bit acting to spread the genes between species quite a lot. Yes?
Wait, so superbugs that are antibiotic resistant, you're saying that no matter what, even in 1,000 years, there's no antibiotic to kill it?
Yeah, so what about the superbugs? Is there no antibiotics to kill them? As much as I know, no. Like, we have developed some new antibiotics in the recent years. Every time that happens, now they make a big paper out of it saying, wow, new antibiotics, but I'm not sure that they are working from the outside, And so what is left for us if there's no antibiotics? Well, we've got immune system, like we can still clear them with a good old immune system, let's not forget that. And second, there's this idea of using actually phage to kill them. And that's an old idea that never really worked, but has also never been tried really, really hard. So what about you find the natural predators of that bacterium and then you just load the body with these natural predators? You could clear the infection this way. It's been done in extreme cases. That's so expensive that this is not the favorite strategy in the Western world. Like, it's more a strategy that's used in the East, in Georgia, places that are kind of in Eastern Europe. They have tried this and they got relatively good, they got good results. But what you have to do to develop these, to find these phages, is to work in the sewers, and so that's so disgusting that nobody wants to do that. So if you don't have to, people prefer not to, but that'd be an idea for what you can do against the superbugs. But that's just an idea.
I just got a quick question. You mentioned the immune system, and I was thinking about the genetic basis to the microbiome. Does that kind of explain the fact that our own immune system doesn't, kill our normal flora? Does that kind of provide an explanation for that genetic basis? Why don't we attack our own?
Yeah, so a great question. Why doesn't our immune system attack our gut microflora? And I'm not sure. So I think it's known. It's just that I don't know it. So if anyone knows here, please tell me. One thing is that the levels of inflammation are really important to control. So when you have an infection, typically SARS-CoV-2, for instance, because people looked at this in detail, the gut microbiome changes a lot. And so, like any infection by a virus, actually. And probably the mechanism is because the level of inflammation rises, some things that used to be tolerated are no longer tolerated. And so there is some sort of a sweet spot where, like in the healthy baseline of immune reaction, there would sometimes happen, skirmishes between the immune system and the bacteria, but overall that makes it stable. But if you tip the balance a bit, then these guys are kicked out. So kind of a hand-wavy answer, but I think that's more or less the mechanism. Yes?
I think I can answer this, but
Go ahead.
this is, I'm taking immunology, taking immunology now, so I can try to answer this. So when you're born, and while you're still in the womb, your immune system is in school. So when it develops, it has particular receptors, like every different immune cells, like neutrophils, macrophages, all the good stuff. And even an adaptive immune system, they have these receptors that would recognize the different carbohydrates and the different glycolipids and all the different things that are on that bacteria. And so okay, this is our workers. We don't need to kill them. Unless you have an autoimmune disease. I'm not sure if there's any autoimmune diseases where they attack the gut microbiota, but generally it goes through a school, and it all has to do with recognizing the receptors. So there's the self and then there's the non-self.
So the answer is that they go to school.
There you go. If I'm wrong, please correct me. But that's how I kind of understand it.
So switching topic a little bit, there's one last high-throughput method that we haven't talked about at all. We've been really focused on sequencing, but there is a very widespread method that you will see also in your papers, which is equally important, which is called mass spectrometry. So we're going to see a little bit how that works. Mass spectrometry is the technology to measure the mass of molecules, as simple as that. It comes in 3 flavors, measuring the mass with time, with angular momentum, or resonance. Either way, you obtain a profile of the compounds present in the sample. Like, you have an idea of their mass. And when you know the mass of molecules, you really have a good idea of what they can be. And so you can measure the time it takes for a molecule to go from point A to point B. You accelerate it in a corridor. And if it's heavy, if the mass is high, the acceleration is going to take more time, of course. If you know a bit of physics, or if you even live in this world, you'll notice that. Another one, which is a little bit more complicated, is the angular momentum. You put a charge on the particles, and if they're heavy, if their mass is high, it's going to be harder to bend them in a magnetic field here. They're going to turn, but they're going to turn a bit slower. Again, it has to do because they're harder to move, because they're heavier. And so the heavy particles that are in orange, red are going to rotate less than the light ones that are in blue. So you'll be able to detect things of a certain mass, things of a different mass. And the last idea that's out there is to take 4 different tubes with different charges, plus and minus, and you keep switching the charges between them extremely fast. And so you're going to create some sort of, zigzagging for the molecules, depending on where you put the charge, because you keep alternating them. And if the molecules are too light, then they're going to actually zigzag so much that they're going to get out of this corridor that you create. If they're too heavy as well, you're going to make it so that they will actually be biased toward one direction. But if they have exactly the mass that you want to select for, they will go through. That's the mass here in green. So you'll see whether something of that exact mass is present in your sample. These are methods of physics. It doesn't really matter to understand them in detail. What matters is that there are many options to measure mass. And if you know the mass, you have an idea of what the molecules are. So with that, with mass spectrometry, you can do proteomics. It's the same as genomics. You measure the proteome, you measure proteins instead of genes. Mass spectrometry is the standard high throughput method to identify proteins in a sample. It identifies peptides, not proteins, and it works better when you know the peptides to look for. Example, through the genome sequence. So here, the idea is that you just purify the proteins out of a sample, and you're going to identify them through their mass. And the way it works is that you cut these proteins into peptides using trypsin. So trypsin cuts, for example, on lysines and arginines. And so that will create short peptides of length average 7, something like that. And if you can identify these peptides of length 7, then you have an idea of what the original protein was. A pretty good idea if you can order them. Okay, so you create these small peptides, and then you're going to use mass spectrometry to measure their mass. And because there's only one way to make exactly one given mass with 7 peptide, then you will know which are the 7 amino acids, the residues in this particular peptide here. Okay, so that's the idea of mass spectrometry. But you see, it works better if you know the sequence of the genome, because if you know the sequence of the genome, you know the sequence of the possible peptides you can encounter. So you can calibrate your method to say, yeah, there are a lot. We're talking of 100,000 or probably more peptides, but they each have a different mass. And the method is just so accurate that when I see that exact mass, I know it's going to be that exact peptide that has to come from exactly that gene. Okay, so that's the idea of combining genome sequencing with proteomics. Tandem mass spectrometry, MSMS or MS2, can be used to sequence peptides based on their mass. So you've got these examples that are here showing what is MS, mass spectrometry. You just pass your samples, and you got peaks here that correspond to different masses. In reality, it's mass charge ratio, because they always charge the particles. And here what you can do is combining 2 mass spectrometry. You have one first mass spectrometry that will select a particular mass, target mass, and then split them again in smaller pieces and then measure the mass again. So you select something for having the particular mass of one peptide that you have identified, but you don't know what this peptide is, and then you chop it by cutting the peptide bonds. So you're going to get all the range of mass from the smallest to the biggest, and by seeing the difference of mass that you obtain, that tells you which is the amino acid that was different between all of these. Okay, just use the mass as the readout for which was the peptide, which was the amino acid that really was cut when I removed it. So that's all based on comparing masses instead of direct sequencing. If you didn't get it because you went too fast, don't worry. All that matters is that there are ways to know which proteins are in a sample. And these ways are all based on mass spectrometry. That's really the standard. So with that, you can do metabolomics. Okay, so that's now the study of all the metabolites in a sample. And LCMS, or liquid chromatography / mass spectrometry, is the standard to identify metabolites in a sample mixture. So you do a chromatography to separate the components based on their size, the general class of mass, something like that, and then you use mass spectrometry to identify exactly what was in each pool. Samples are first separated by chromatography and then analyzed by mass spectrometry. The obtained profiles are matched against a reference of known metabolites to identify those that are present in the sample. So you got here T1, T2, T3, T4. These are different fractions that have been obtained by chromatography, and then each of them is run on the mass spectrometry, and you get a profile that really tells you what are the possible compounds present in each fraction. And then you have a reference of metabolites, so you can tell, oh well, this thing here with that exact mass, it must be CO2, whatever you're looking for. Not all metabolites can be discovered. Example, if their abundance is low or if they're not expected, okay, they're just not in your reference, you're not going to get a match. But if you have a good idea of what you're looking for and it is present in your sample, then you're going to find it with this method. You're going to get a good match for when it was supposed to go in the liquid chromatography and what is the mass that you expect for the mass spectrometry. So the human metabolome is really everything that we can find, the metabolites that we can find in the human body. And remember, but I think you got the point already, many of them are not produced by us. Many of them are produced by our gut microbiome. The OTUs in the gut microbiome produce and consume metabolites that diffuse through the blood to the rest of the organism. The metabolites in the human body thus depend on factors influencing the gut microbiome: genetics, diet, medication, chance. Okay, so this here shows that the metabolites that are produced in the gut will go to the blood and then cross the blood-brain barrier, go to the brain, potentially affect our behavior, whatever that is. Okay, everything is like is a whole, of course, that's a body, and you got a bit of some of the chemical reactions happening in the gut because this is where the bacteria are, but the metabolites they produce will diffuse in all the body. So that's all I wanted to say for today. The key points to remember is that metagenomics gives access to uncultured microorganisms. Okay, the one thing you have to remember about metagenomics is that it allows you to know something about the bacteria that we cannot culture. Otherwise, we just don't have another method. The viruses are strongly underrepresented because we don't really know what we're looking for, and so that means that we don't find it, of course. And finally, the gut microbiome is important for health and also for diagnostic. And so we were supposed to have some breakout group activities, but we're not going to have them. Now there's no time. So we have 7 minutes for questions. So go ahead, really don't be shy. We have plenty of time. And at the very end, I would like to keep 1 minute to ask your opinion about how the session went in person and whether we can change something for next time. But for now, just ask questions about the class of today, if any. Otherwise, going to be 7 long minutes of silence. Yes, Maureen.
Hi, sir. I have a question in regards to the blog post. Can we write like importance of it or like critique or technically or opinions?
Okay, so the question is about the blog post. Are we allowed to write an opinion and to make a critique? Yeah, of course. All right. So whatever is, like I said, the point is got to be interesting. That's the most important by far. The second most important is got to be accurate. It's going to have some science in it. So if you just say, well, this article, I don't like it. I'm not, it's not going to carry it, like definitely. But it's also maybe because it's not very interesting if it's the only thing it contains. But if you first say what they say is that, and that's really what they mean, but these are other things that have been said, and these are other things that are conflicting or that are not well done, that's perfectly fine. It's called science, actually. That's what you're supposed to do. You're supposed to say, these are the results, and we're not even discussing whether they're real or not. They're facts. Now, there may be missing controls. There may be not having checked for some swap, not having checked for whatever that is. So you can criticize that. Or you could say, they interpret their result this way, but they also don't interpret it this way, which is a frequent interpretation. That's totally fine, right? So the answer is yes, you can criticize. Should you? It's entirely up to you. So far, have a look at the Genomic Odyssey. That's a very good example of what people have done. You will see that people were relatively neutral. And also the other thing that you have to know is that I will ask you to contact the authors of the studies. So if you can get their opinion of the article, it's even better. Sometimes you will see that they will tell you something about, oh yeah, sure, it was hard because actually one of the guys just left the study in the middle because he found a job, and so we had to compensate for this, and that's why it looks a bit weird. Sometimes you're going to get interesting information from the inside by just talking to the authors. And you will actually see that these are opinions. They're not facts anymore, what people think about whatever happened. So the answer is yes, totally you can, and you don't have to. That really is up to you, but you should not be shy. You can put your opinion, your perspective. That's expected. Other questions? About the blog post, about the content of today? Yes.
So I have a question about the max stack. So because I know from the other course, like the max stack can also be used to identify not only the peptide sequence but also like the translational modification. So I'm just wondering, like when we bombard like the fragments, so we already break like the polysaccharide modification from the peptide, or they're still connected together, and then we can I'm just like wondering, because there are so many diverse kind of like PDM, that how can we just, and then their combination with peptide will be even more diverse, like so difficult to like recognize.
Yeah. So the question is about post-translational modification. Like mass spectrometry is especially good for recognizing post-translational modifications, especially of histones, because histones have many and they're important, but not just histones. So how does that work? The answer is it works exactly the same. It was just fine. Each of these modifications has an exact mass, and these methods are so accurate that they can totally distinguish them. They can even totally distinguish 2 or 3 modifications at the same time. Right? Like the atoms, every atom has a different mass. And just the difference of 1 hydrogen, which is tiny, 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 is very visible from mass spectrometry. It's super easy to pick up, just 1 hydrogen. So for these methods, like the chance that 2 modifications on a peptide would have exactly the same mass are actually quite small, so that's enough to identify them. On the same protein, yeah, that'd be too big, because now the combinations become way too many. But that's not what you do. You chunk them in smaller pieces first, 5 to 7 amino acids, and there usually within this 5 to 7 amino acids, you don't have 100 modifications, you have 1, maybe 2. So that's how we can do it. Okay, so now it's time for, let's say, a quick debrief. So people in the room, how did that go today for you? Like, put a big thumbs down to say this was really bad and you were totally bored, and a thumbs up to say it was okay. So some thumbs up, that's good. Is there anything that is obviously improvable from your point of view? Not obviously. You need to think about it, if anything. People in the online, in the last minute that we have in the chat, tell me if there's anything that really did not work well for you today and that we have to improve for next time. And people in the recording, for you as well. Send me an email. You guys, please send me an email saying what didn't work for you in the recording. Okay, there's nothing in the chat, so I guess people need time to think about it. And we need to evacuate now because there's a class. So thank you very much. See you soon.